The next question is, which one of the following insecticide is likely to cause phytotoxicity on onion? What is phytotoxicity? It is like a chemical injury to the plant caused by the pesticide, especially. Okay, and also the fertilizers. You can tell insecticides means that what is the insecticide? It is mainly the chemical to kill the insect, right? So there's a chemical injury to the plant mainly caused by the insecticide. Okay, so on the onion it shows up, leaf scorching and tip burning, and likely to have the yellowing, and bulb size will and all will finally get reduced. Okay, so onion leaves generally have a waxy coating, right? But they are very thin and sensitive, so the insecticides can easily burn them away. So they asked which insecticide is likely to cause the phytotoxicity. So the options are monocrotophos, imidacloprid, dimethiot, profenophos, and answer is not known. And the correct answer is profenophos, which is most likely to cause phytotoxicity, and this is the organophosphate insecticide. Okay, and this is the insecticide as well acaricide. Acaricide means this particular insecticide is used to kill the mites also. Okay, and it is labeled as phytotoxic to onion in ICAR extension guides and all. So profenophos is the phytotoxic, especially to onion, because organophosphate insecticides, they have a high solubility, and emulsifiable like EC concentration will be there. No, emulsifiable concentrate formulation can easily dissolve the waxy cuticle or the waxy layer on the onion leaf. So this will lead to the direct chemical burn, especially at the higher dose. Okay, and also the higher temperature. So that's why you will often see the warnings like do not use on onion, crucifers and all, especially on the profenophos labels. Okay, and why not the monocrotophos? Means it is a moderate risk only, but it is less than the profenophos. And this is also the organophosphate, but it poses the moderate risk, not that much risk caused by the profenophos. Okay, and imidacloprid, this is also having the low risk, and this is again the neonicotinoid, systemic insecticide, not the organophosphate one. It is generally, considered as a safe for the onion when used at the recommended dosage and all. Why, it is generally absorbed by the plant and doesn't sit on the leaf surface to cause the burning and all, unlike profenophos. So profenophos will directly sit on the leaf surface of the onion and will degrade the wax cuticle, and completely it will cause the burning symptom. Okay, and that's why this imidacloprid can be recommended for thrips control in onion. And dimethiot, again it is organophosphate, but it is possessing the moderate risk. Okay, and the next question is the ETL for leaf infesting mite in cotton. So what is ETL? This is the economic threshold level, the pest density at which the control measures can be applied to prevent the particular pest population from reaching the economic injury level. Economic injury level means there will be the drastic yield loss and we cannot even face it, right? Here the leaf infesting mite in cotton means usually refers Tetronychus utricate. Okay, this is the two-spotted spider mite, usually can be seen in the cotton, Tetronychus utricate. And also the red spider mite will also come, Tetronychus cinnabendus. So this will generally feed on the underside of the leaf of the cotton, and this will cause the yellowing, and eventually leaf drop will occur. So the options are the general ETL for this mite in cotton is the options given for this are 5 per centimeter square, 10 per centimeter square, 15 per centimeter square, 20 per centimeter square, and the answer is not. Means that if there is 5 per centimeter means that much only allowed. So at the stage we should take the control measures in order to prevent the infestation caused by the mite. if you are not taking enough action, it will reach the EAL. Means that there will be the complete or drastic yield loss will occur. Okay. So these are all the options given for this. And the correct answer for this is 10 per centimeter square. So as per the ICAR guidelines and CABRC extension material, the ETL for the mite on the cotton is 10 mites can be allowed per centimeter of leaf area. Okay, so per centimeter of leaf area, only 10 mites should be there. So at this stage, we should take the control measure. So that's why we can prevent them like to reach the EAL. Okay, means that you count the mites on the undersurface of 20 to 30 randomly selected leaves from the middle canopy. You have to select the 20 to 30 leaves in the middle field area of cotton and calculate the average per centimeter square. If it is more than 10 means the spray is advised. And if it is reached 10 means the spray can also be advised. So at this density, feeding damage starts reducing the photosynthetic area significantly. So yield loss will become, highly economical. And 5 and all this is again still below the damaging level. So this is not applicable for this. And 15 means it is too high. So by the time you hit 15 mites per centimeter, no, like centimeter square, damage is already visible and yield loss will already started. Yield loss has already started. So this is closer to the economic injury level, not the threshold level. Okay. And the same 20 is also too high. So the correct thing is 10 per centimeter square. And also, the certain things you should remember. So whenever you are going to take a sample, check third to fifth leaf from the top. Okay, top underside using the 1 centimeter area. And generally the mites outbreak will be more in the hotter, the dry regions. Okay, especially 2 to 3 months after sowing in cotton. So at this stage, what we should do means we should use the acaricides like dicofol, proporcate, or abamectin and all. And also you should remember one thing, we should avoid the synthetic pyrethroids. Okay, because as they flare up the mites, they will do the do resurgence. The population will move. Okay, so better to avoid the synthetic pyrethroid. Go for the better acaricides like dicofol, proporcates and all. And the next question is in rice Tungro virus endemic area, the ETL, that is economic threshold level for the rice green hopper is they ask. You know what is ETL and all. So generally for rice green leaf hopper, which is Nephrotitis presence, the ETL is different in the rice Tungro virus endemic area. Why? Because this is a vector, right? GLH is a vector for the particular virus. Even the small or few virulliferous virus can able to spread the virus and they can cause the massive yield loss. So the threshold should be kept very low to prevent the virus spread. Okay, and the different options are given, and those are 5 per hill, 3 per hill, 2 per hill, 6 per hill. And the answer is not known. And the correct answer is 2 per hill. This is correct for the Tungro endemic area. So in Tungro endemic area, as per the guidelines given by CABRC, ICAR and all, ETL for GLH is 2. Okay, per hill. So at 2 hoppers can be allowed per hill. So the risk of virus transmission becomes high. Okay, so at this stage, the risk of virus transmission becomes high. So the goal is to kill the vector before it spread the rice Tungro virus. So this is for the vegetative stage up to 45 days after transplanting when the rice is more susceptible to Tungro. So when it is susceptible to Tungro means after 45 days of transplantation. Okay. And other options are not opt for this question. Either it may be high or it may be low. In most of the cases, it is high. It means that in this case, the complete field will be devastated. If we allow this much 5, 6, 3 means it is more than the yield, right? So definitely there will be the yield loss. Okay, so better for the rice leaf hopper in rice to grow virus endemic area 2 per hill is allowed. The next question is which one of the following insecticide is more safe to honeybees? Why honeybee safety matters means that insecticides are generally rated by their toxicity, right? So most of the insecticides are posing toxicity to the honeybees based on the LD50 values and field values and all. Safe to honeybees means that generally they may pose the low direct toxicity. The toxicity effect will be very low, or there will be the low residual activity can be found on the flowers. Okay, so spraying toxic insecticides during the flowering stage kills the foraging bees and all and affect the pollination. Okay, safe means those particular insecticides are possessing the low toxicity effect. Okay, so the options given for this are diflubenzuron, chlorpyrifos, cypermethrin, imidacloprid, and the answer is not known. And the correct answer is diflubenzuron. This is mostly safe to honeybees. Why means this particular insecticide belongs to the chemical classes, insect growth regulator, that is benzoyl urease group. Benzoyl urease group. Okay, this is IGR, insect growth regulator. Mostly it inhibits the chitin synthesis. Okay, so this particular insecticide affects only the insects during the molting or larval stage. So adult bees generally don't molt, right? So it has very low direct toxicity to the adult honeybees. Okay, so here the bee toxicity is classified as practically non-toxic. Practically non-toxic means LD50 values more than 100 microgram per bee. So that and all in advanced state. So as of now, you should know this is posing the very low toxicity. Reason it doesn't kill the adult bees on contact. When it is coming in close with the insecticide, it won't kill because it is not a neurotoxin. So it's mainly used for killing the caterpillars and larvae and all. Okay, so it can affect the bee larva. Okay, if brought into the hive in pollen and all, but field risk is generally much lower than the hive like neurotoxic insecticides and all. Okay, and why others are wrong means chlorpyrifos, it is also posing the high toxic. So this is the chlorpyrifos organophosphate insecticide. This is posing the high toxic to bees. Okay, this is the broad spectrum neurotoxin. This is extremely toxic, less than 2 microgram per bee. Even the low dose can, able to kill the bees on contact as well as through residues. Okay, and cypermethrin is again the synthetic pyrethroid group and having the neurotoxin effect, and again it is having more or highly toxic to bees. Extremely toxic. Again, it is having the less than 2 microgram per bee LD50. Why? Because this is causing the fast knockdown effect. Means that immediately it will, express their toxicity in their body. Most of the pyrethroids carry the danger to the bees that is mentioned in the label. And imidacloprid, again, it is coming under the neonicotinoid group, systemic neurotoxin, and this is also possessing the high toxicity. LD50 is again less than 2 microgram per, bee and all. Okay, because it is systemic nature means that it easily moves into the nectar and pollen of the plant, and this is linked to having the sublethal effect in the bees. So this neonicotinoids and all banned in the most of the countries. So the correct answer is diflubenzuron, which is the insect growth regulator. And the next question is to prevent the spread of which pest in Tamil Nadu, quarantine stations were started at Mettupalayam, Kudalur and Shenbaganur during 1943. So what happened in 1943? There is a Madras presidency started the quarantine stations at Mettupalayam, Kudalur, Shenbaganur. These areas they started the quarantine station just to stop the spread of one particular insect. Okay, That is what we have to find here, because this was a planned quarantine measure that was taken under Destructive Insect and Pest Act 1918. So the main goal of the particular stations was to prevent the pest from moving from the one area to another area. Okay. So the options given for this are Viriosoma lanigram, that is woolly aphid on apple, Thorima opercola, which is nothing but potato, tuber moth and potato, Isaria pachisi, which is the cotton cushion scale on water, Coccus vividus on coffee, that is coffee scale. Okay, and finally the answer is not known. And the correct answer is Isaria purchasey on wattle. So the common name is cotton cushion scale. So the host for this particular pest or wattle, wattle means acacia, okay, but also it may be seen on citrus, rose, and many other plants. Okay, so how it causes the damage means that it usually sucks the sap. Okay, suck the sap from the leaves and almost all area and causes the sooty mold. Sooty mold will be there. There will be the developmental black color fungus over the leaf area. So this will weaken the trees and reduces the bark yield and all. Because at the time wattle was a major cash crop in Nilgiris and all for tannin production, right? So at the time, so in order to product the particular crop from this pest, they established the quarantine station at Mettapalayam, Gudalur, Shenbaganur. Okay, it is a highly invasive pest. Once it is introduced, it spreads fast through the crawlers and the wind also. It can be easily carried out through the wind. So quarantine was the main control before the biological control with the Rhodolia cardinalis. So in order to control this pest, Rhodolia cardinalis was imported from foreign country. Rhodolia cardinalis is the Vedalia beetle. It is like a biological control for this pest. That also you should know. Why others are wrong means Iriosoma lanigram, which is the woolly aphid. This is mainly be occurring in the apple and all in Himachal, Jammu and Kashmir and all. So quarantine measures for this were started later, but not in the 1943. Thorima operculla, it is a potato tuber month. So for this, quarantine measures has been taken in Kodaikanal and Nilgiris areas. Okay, but the majority actions was in 1950s and 60s after it became the serious issue, not in the 1943. Okay, and Coccus viridis on coffee, it is a green scale, right? So the control was through the field management, not through the quarantine station and all. So the correct answer is Isaria purchasing on wattle. Okay. So why these three places means Mettapalayam, Gudalur, Shenbaganur are the entry points, right, to the Nilgiri hills from the plains. So they were used as a check post, okay, to inspect the wattle planting material and prevent this particular pest from the spreading downward. Okay. The next question is effectiveness of the Bacillus thuringiensis toxin will be more in pH. So how this Bacillus thuringiensis or we can tell BT toxin works means this particular microorganism produce the cry proteins, right, during its florulation time. So those proteins are generally inactive one. So they are called protoxins. But like when they are ingested by the insects, this particular protein will be the proactive stage, which is nothing but the inactive stage, right? But the effectiveness only depends on what happens inside the insect gut. What pH will helps to make the inactive protein into active protein. Okay. So the options given for this are alkali, acidity, neutral, no relation, and answer is not. So generally what happen? Insect eat the BT, right, and crystals. Why? Because this is the insecticide, right? So BT can be applied to control many of the lepidopteran insects. So it is applied, and the protoxin or the proactive one will be dissolved in the insect gut. Okay, and there is a certain pH will helps to make the protoxin into active toxin. so that the toxin will bind to the midgut receptors of insect. And since they bind into the midgut receptors, there will be the formation of pore. Okay, so that's why it causes the gut lysis and death and all. Okay, so which pH we have to find? That is the question here. And the answer is alkali. Yeah, midgut of the most of the Lepidopteran larvae is highly alkaline. Usually, pH 9 to 11, more than 9. So what happened in alkaline pH? The cryprotoxins generally solubilized easily. Okay, if the pH is acidic or neutral, means that the solubilization will not occur. The solubilization will poor. So since it is an alkali pH, the solubilization will be easy. Then what happened? This alkaline condition also make the favorable condition for the activation of like Bt toxin. So the inactive stage will be turned into active one. And also it active the gut proteases like trypsin, chymotrypsin and all. Other protease will also be activated because of this alkaline pH. So that's why BT is mostly effective against caterpillars whose gut pH is alkaline. Acidity and all usually 4 to 6. This will prevent the solubilization. Okay, and the same neutral, this and all doesn't work with the BT. Okay, so in this case, BT is ineffective for the lepidopterans. So only when they are in the alkaline stage, BT will work. Okay, the next question is, in artificial beehives, bee space or bee passage was discovered by. So this one we have already discussed, but there the question is different, right? Like there the question is related to modern beekeeping and all. Okay, here they ask bee space or bee passage was discovered by. What is bee space? It is a gap, right, around 6 to 8 mm that bees can easily leave between the home. Okay, so if the space is too smaller than the 6 to 8 mm means bees can seal it with propolis. Okay, if it is larger means they fill it with the foam. So that is uncomfortable for the bees to move around. So at 6 to 8 mm, where the bees use it as a passageway and they don't block their work. So that is the correct space. So the discovery made possible only the introduction of mobile frame hive. So who discovered it? They asked. So the options are Beeson, L. Langstroth, Newton, Abbe, and the answer is not known. And the correct answer is Langstroth. That is Lawrence Lorraine Langstroth. He is an American apiarist, discovered the bee space in 1851. Okay, and while inspecting the hive, he noticed bees left only very small gap around the home. So he standardized the space. He used the principle to invent the Langstroth mobile frame hive in 1852. So this was the breakthrough, because before that beekeepers had to destroy the complete colony in order to harvest the honey, right? So if we provide the proper space means we can directly take the frame out of the box and we can easily keep it in the honey extractor chamber, and after extracting the honey we can keep it back. So here the home destruction will not happen. We can save our home. So we can reuse it also. Okay. And Beeson, he was a Indian entomologist and worked for forest entomologist. So he didn't discover the bee space and all. And Newton, he had nothing to do with apiculture. And Abby, he was developed the very high, but he used the principle of this bee space discovered by this person Langstroth, but he didn't discover anything related to the apiculture. So the correct answer is Langstroth. The next question is Toy sac brood virus specifically attack. So this is a particular virus. It is a picorna-like virus that is causes the sac brood diseases and honeybee brood. So here what happen if the bee larva is infected with this particular virus means those larvae will fail to become a pupa. So the body will completely be filled with a fluid and they die in the sac-like stage. So that the name comes sac. So it's spread mainly through the contaminated food, beekeeping equipment, and robbing behavior and all. So this particular virus mainly attack which honeybee species they asked. And the options are Apis mellifera, which is an Italian bee or European bee, Apis florea, which is a little bee, and Apis serrana indica, which is an Indian bee, Apis dorsata, that is a rock bee. And the answer is not. So the correct answer is Apis serrana indica. Because the Toysat group virus generally attack the Apis serrana indica, that is the Indian hive bee. It was first identified as a major problem in Thailand, especially in the 1970s, where it caused the massive colony losses in Apis serrana. So that's why it is named Thai. Thailand means Thai. Sac means it forms the sac-like structure, and brood means it is generally affect the brood, the young one of the bees, not the adult. So Thai sac brood. Okay. And in India, this Toysac brood virus outbreak occurred at, late 1980s, 1990s especially. So this one was wiped out most of the Indian bee colonies in Kerala, Karnataka, Tamil Nadu and all. Because, our Apis serrana indica has very low resistance to the Thai strains, virus strain, which is why the disease is so devastating one. Okay, and Apis mellifera, it is somewhat resistant to the particular virus strain. It can carry the virus, but rarely shows the symptom. So that's why Apis mellifera was introduced in India after the Apis serrana colonies collapsed. In order to avoid the infestation caused by the virus, this Apis mellifera was introduced into India. Okay, and Apis florea, it is a dwarf honeybee. It is not a primary host for this particular virus. And Apis dorsata, it is a rock bee. It is not at all susceptible to the particular virus under the natural condition. Why it is matter in India means before 1990s, the Apis serrana, our Indian bee colony was a major species. Our Indian bee was the major species used for the traditional beekeeping, right? But after this virus comes, the 80 to 90% colony loss was seen in many regions, which led to the shift to the Apis mellifera beekeeping. So now the Apis mellifera recovering in some areas due to selective breeding or resistance thing. Okay, because it is highly susceptible to the virus. And the next question is the dammer bee home is made up of dark material called. So that is a mixture of wax and earth or resin. So the material they asked, dammer bee, which is also known as mosquito bee. Okay, Tetragonella species or Melipona species, in which the home is made up of certain material they asked. But the material is mixture of wax and earth or resin. Okay, so the options are bee bread, propolis, royal jelly, cerumen, and the answer is not known. And the correct answer is cerumen, which is generally, dark sticky material used by the stingless bee, which is also known as mosquito bee, dammer bee. Okay, and this is the mixture of bee wax, plant resin, or propolis, earth, mud, and all. So the color may be varied from dark brown to black, which is why it looks different from the light yellow wax of AP species. So generally bees collect the resins from the trees, right? So those resins will be mixed with the wax secreted by the bees, right? And that by using the material they hope or they construct the home. Okay. And why others are wrong means bee bread, which is the fermented pollen, right? Fermented pollen stored in the cells. Bee bread means pollen plus nectar or honey. That is like a feeding material for the young ones, and that is not having any relevance with the dammer bee home material. And propolis, which is a resin-like substance collected from the trees. Okay, and royal jelly, it is a protein-rich substance, mainly secreted from the nurse bees, that is a worker bees, and this will be given to the young larva as well as queen. So this is not having any relevance with the cerumen and all. Okay, and the correct answer is cerumen.